In this video, we're going to take a closer look at the conifer life cycle, and we're using this as an example of gymnosperms in their life cycle. Like all plants, conifers and gymnosperms exhibit the alternation of generations. That includes both gametophyte and sporophyte generations. There are some specialized structures and terms associated with the conifer life cycle, however. Conifers are heterosporous. Hetero means other or different. And this just means that there are two different types of spores produced by conifers. This means that there are essentially male and female gametophytes. So we've got a male and female cone, and these cones are also known as strabili. And these are the gametophytes. So the heterosporous means that there are male cones and there are female cones. And you can see those differences here on the picture. So these are the female cones, the ovulate cones. And these are the male cones, also known as the pollen cones. Now, conifers are monoecious, and mono means one. So they do still have male and female cones. The one comes from the fact that these cones are on the same plant. So one plant has the male and female gametophytes present. So you can see this tree here, this sporophyte, and it has both the female cones and the male cones present on one tree. So one tree gives you the monoecious designation, but having two different cones forming two different types of spores gives you the heterosporous condition. Let's take a closer look at the female cones. So the female cones are known as ovulate cones, and they are larger than the pollen cones. Remember that strabili or strabili just means cone. So you can see here that this is the strabilis or the cone, and this cone is made up of scales. So each one of these pieces right here is a scale. Each scale is a sporophyll, and if you remember when we talked about the seedless vascular plants and ferns, we talked about these modified leaves that produce spores. So these scales don't look like a typical leaf, but it is a modified leaf and it produces spores. Now we can take a closer look at an individual scale down here in this picture. And each scale has two megasporangia. And the megasporangia are essentially the female structures that produce the gametes. A megasporangium includes an ovule surrounded by an integument or a coat, kind of a protective layer around it. And this picture doesn't show a ton of detail, but you can see the ovule present here. So that is a megasporangium. The megasporangia contain megasporocytes, which we will talk about in more detail on the following slide. As we mentioned on the previous slide, the megasporangia contains megasporocytes. So within the ovule, we have these megasporocytes. And these are diploid cells, so they have two sets of chromosomes, and they're going to go through meiosis. And meiosis is the process that can take a diploid cell and essentially make haploid daughter cells. So this megasporocyte goes through meiosis to form 4 haploid female megaspores, and these are the daughter cells of meiosis. Now 3 of these cells degenerate, so these are useless, but one survives, and this is typically the bigger, stronger of the daughter cells. Now, the surviving megaspore is going to go through mitosis. Remember that the megaspore is haploid. In mitosis, the ploidy stays the same. So the haploid cell is going to produce additional haploid cells during mitosis. And this is ultimately going to form a multicellular haploid gametophyte. So we said that the cones were the gametophytes, and really the cones contain the gametophytes because the female gametophytes are forming inside the scales of the cones. Each ovule in these scales has an opening called a micropyle, and it's not labeled on this picture here, but we'll go ahead and label it now. So this is the micropyle. And pretend like the pollen grain and pollen tube are not here yet. So the ovule has this micropyle that develops a sticky pollination drop. And this helps to trap pollen. So when pollen sticks to that pollination drop, it starts to form this pollen tube, which ultimately delivers the male gamete to the female gamete. So this delivers the sperm from the pollen to the egg in the ovule. Remember that conifers are heterosporous, so they do have male cones that are different from the female cones. So the male cones are also known as pollen cones, and they are smaller cones. Remember, strabili or strabili is just a fancy word for the cone. And these cones are made up of scales just like the female cones are. So you can see right here, this is one scale. And just like the female cones, each scale is just a sporophyll, which is a modified leaf that produces spores. The scales have microsporangia, which are also known as pollen sacs, and these structures contain microsporocytes. And we'll talk about the microsporocytes in more detail on the next slide. So within each scale of a pollen cone, there are going to be these microsporangia. And within the microsporangia, there are going to be these diploid microsporocytes. And these diploid microsporocytes go through meiosis. So remember, in meiosis, the diploid condition ends up producing haploid daughter cells. And so 4 haploid male microspores are produced during meiosis. These microspores go through mitosis, which means that their ploidy is going to stay the same. So remember, haploid cells produce other haploid cells in mitosis. And this forms the pollen grains, which are known to be the mature male gametophyte. So this is the gametophyte generation of the alternation of generations. So remember, the microsporocytes are the cells that are producing the microspores, and then the microspores produce the pollen grain. So let's take a closer look at one of these microsporocytes. We're going to kind of zoom in here and kind of jump ahead and take a look at a single pollen grain. So a pollen grain has a tough coating around it, and this is known as the pollen coat. And this protects the cells inside. Inside this pollen grain, there are 2 cells. And each cell has a haploid nucleus in it. One of these cells is a vegetative cell. Which is also sometimes called a tube cell. because this cell is going to be responsible for forming the pollination tube after pollination occurs. So when this pollen grain lands on the sticky pollination substance at the micropyle of the female scale, the vegetative cell is going to produce a tube that leads from the micropyle to the ovule. The other cell is a reproductive cell. Because after pollination, it is going to fertilize the female gametophyte. So the reproductive cell is actually going to be involved in fertilization in order to form a diploid zygote. As we mentioned on the previous slide, a grain of pollen, so I'll just write pollen grain here. So each pollen grain contains one haploid vegetative cell, also known as a tube cell, and one reproductive cell. Remember, the tube cell is ultimately going to form the pollen tube to deliver the reproductive cell to the female gametophyte so that it can fertilize the female gamete. The pollen grain is encased in a tough tissue for protection. This is also called the pollen coat. And the pollen gets released from the megasporangia, which are in the cones. So if we look at this picture here, we can see the cones here. And this person is just moving part of the plant around with their hand. And you can see all of this kind of hazy looking stuff here is the pollen. And this pollen is typically carried by the wind to either fertilize female cones on the same plant or fertilize female cones on surrounding plants. You may be familiar with the kind of this event of the pollinating that occurs in the spring, especially here in South Carolina, where the air can actually look kind of hazy because there's so much pollen in it. And that pollen settles on things like your car and it kind of coats it in this yellow pollen haze. And that pollen is basically plant sperm that is trying to be dispersed to go find a female plant to fertilize. So now let's talk about what happens during pollination, and make sure you don't get pollination and fertilization confused. So pollination and fertilization are not the same thing. Pollination is where the pollen lands on a female structure and delivers the male gamete to the female gamete. Fertilization occurs when the male gamete actually fuses with the female gamete. So during pollination, remember that in our female scale here, there is this opening known as the micropyle. And this micropyle has that sticky pollination drop. And so if pollen is in the air, it might stick to that pollination drop at the micropyle. And remember that a pollen tube has two different types of cells in it. The vegetative cell, also known as the tube cell, creates a pollen tube that enters the megasporangium. So you can kind of see that here where you've got your pollen grain. And inside that pollen grain, you have your vegetative cell and you have your reproductive cell. So the vegetative cell is going to start creating this pollen tube here to deliver the reproductive cell to the ovule. And this is where the female gamete is. The reproductive cell in the pollen grain is going to go through mitosis to form the male gamete, which is a haploid sperm. So remember, the pollen is the male gametophyte. Which is the structure that produces the male gamete of sperm. The sperm then travel down the pollen tube towards the megasporangium and the ovule, which is where the egg is waiting for it. So now let's take a look at what happens during fertilization. So pollination has already occurred, and the male gamete, the sperm, has been delivered to the ovule of the female megasporangium. So during fertilization, a haploid cell, in this case the sperm, which is the male gamete, its nucleus fuses or unites with the haploid nucleus of an egg. I'm going to remove fertilization here because I put it here instead. So essentially with fertilization, you are taking a haploid nucleus, fusing it with another haploid nucleus in order to form a diploid nucleus in a single cell. So the fusion of egg and sperm produces a unicellular, so single-celled, diploid, so it's got those two sets of chromosomes, one from the sperm, one from the egg, and this forms a structure known as a zygote. So a single cell produced by fertilization is known as a zygote. That zygote is then going to go through mitosis, so those diploid cells are going to produce more diploid cells in order to form an embryo. And an embryo is basically a developing baby plant. So let's kind of draw that out down here. So you have the egg cell, which has a nucleus that is haploid. And you have the sperm cell, which also has a haploid nucleus in it. And the sperm fuses or fertilizes the egg. So it fuses with the egg. And this produces a single cell with a diploid nucleus that's known as the zygote. And then that zygote is going to go through mitosis to form daughter cells, and these daughter cells are all going to have that same diploid condition. One cell is going to form two cells produces four cells produces 8 cells. And these multiple rounds of mitosis is known as embryonic development. So the structure here is a developing embryo. And this process is going to keep going, and the number of cells are going to double. So you're going to end up with more and more cells. A seed coat is then going to develop around that embryo, and the seed coat comes from the female tissues of the female gametophyte. Now, seed development is a really slow process in conifers, and it might take up to 2 years in order to actually produce the seeds. So after a seed is produced, it is released from the ovulate cones. Remember, the ovulate cones are just the female strabili. And if the seed lands in a favorable environment, germination will occur. And germination is just mitosis. So the embryo inside the seeds, remember that the seeds here have a seed coat with a developing embryo inside. And so this embryo is still going through mitosis over and over again. Eventually that seed coat is going to break down. The cells in the embryo are going to start to differentiate, and ultimately it's going to form a new sporophyte. And what you're seeing here, basically this entire tree here, is the sporophyte of the conifers. And then remember that the cones on the sporophyte contain the gametophyte generation.